We're going to take a look at the chemical class of chemical messengers, so what they're made of. We're going to start with the amino acids. So remember, the amino acids are hydrophilic. So their receptors are going to be located on the cell surface, so on the target cell membrane. And the reason that is because they're hydrophilic, they can't diffuse across the cell membrane and get inside the target cell. So it'd be really dumb to have receptors inside the cytoplasm or the nucleus because they wouldn't be able to get there. All of the amino acid messengers are neurotransmitters, and we're going to come back and take a look at these in Chapter 8, so these will reappear later. So because they're all neurotransmitters, they're going to be made by neurons, and they're going to be stored in vesicles, little membrane-bound sacs. And when the neuron gets the signal to synapse with the next cell, they will release those vesicles by exocytosis. And there's 4 different amino acid neurotransmitters, so they're all made from amino acids. So we have glutamate and aspartate. Both of these are excitatory. And then glycine and GABA, these are inhibitory. And all of these amino acid neurotransmitters are going to be used in the central nervous system, so brain and spinal cord. And this is just a picture of the amino acid neurotransmitters, the 4 of them. And we recognize an amino acid because remember, amino acids have the amino group, a carboxyl group attached to that carbon, and then they have that R side chain that makes them unique. The amines are made from amino acids, and because they're made from amino acids, they're going to be hydrophilic except for, and this is a big exception, the thyroid hormones. And the thyroid hormones we're going to talk about in Chapter 6, and these would be T3 and T4. There's 2 of them, and that's what they're called. The amines, how they're made from the amino acids, so if you have an amino acid here with the amino group, there's an R side chain, and then here's that carboxyl group. To make an amine, and you don't have to know this, but this is just how it's made, all that happens is that that carboxyl group gets chopped off. And then what's left, that's an amine. So that's where it comes from. So you can see that it's certainly made from amino acids. And then answer your question, we're going to answer this. So amines are made from amino acids. And because the amines, except for thyroid hormones, are hydrophilic, their target cell receptors are going to be on the cell surface, so in the plasma membrane of the target cell, because they can't diffuse and get inside the cell, so it'd be pretty dumb to have receptors in the cytoplasm or the nucleus. They're going to be stored in vesicles, and then they're going to be released by exocytosis. And there's lots of different amines. Some of them are neurotransmitters, some are hormones, and some are paracrines. You don't need to memorize that part. So you do need to memorize the different amines, and I did put down which amino acid they're made from. You do not have to memorize that. Okay, so I don't care that you know that catecholamines are made from tyrosine. I just care that you know that they're an amine and they're made from amino acids. Okay, very important to memorize this group called catecholamines. And we'll show you a picture of why they're called that in a minute. And we have 3 of them. We have dopamine, norepinephrine, also called noradrenaline, and epinephrine or adrenaline. So those are the 3 catecholamines. So they are very similar in their structure. They have a catechol ring that we'll show you a picture of. And then the thyroid hormones, those are amines, but remember they're hydrophobic instead of being hydrophilic, but they are amines. And then we have histamine, made by some of our white blood cells and also used as a neurotransmitter in the central nervous system. And then we have serotonin, another neurotransmitter, and melatonin, a hormone. So some of these are really easy to recognize that they're amines because their name, like dopamine, ends in amine. So you know for sure that they're an amine. And then the other ones you'll just have to memorize. And this just shows you the catecholamines. So we had dopamine, norepinephrine, and epinephrine. And so what makes them all catecholamines is they have this chemical structure called a catechol group. So they have this ring here, the catechol group, and so all of them will share that in common. And then it just depends on the cell which enzyme they have to determine which one of these that they're going to be making. All right, and then we get to our peptide and protein messengers. So out of our chemical classes of messenger, this is the biggest group. So if you're not sure what the chemical messenger is made out of, protein is a pretty good guess because there's a lot of protein chemical messengers. And because proteins are made of amino acids, they are hydrophilic. There is a difference between peptide and protein chemical messengers, but I'm not going to distinguish between the two. It's just based on the length, how many amino acids they have. So usually I'll just say protein, or sometimes I'll say peptide chemical messenger, but I'm just, talking about the same thing. I'm not going to distinguish on the length. And because the protein messengers are hydrophilic, the target cell receptors are going to be located on the cell surface, because they're not able to diffuse into the target cell. And some of these protein messengers are neurotransmitters, some are hormones, some are paracrines. You don't have to memorize that. And this is just to show you some of the protein hormones. And you can see that they're a large group. So you can see why I said that if you're in doubt, what the messenger is made out of, what chemical class it belongs to. If you say protein, you get a pretty good chance because that's our biggest group. And we'll talk about a lot of these hormones later on in Chapter 6. All right, so how the protein messengers are made, they're going to start off as a larger protein called a pre-propeptide, and then pieces get cut off, and then it becomes a propeptide. And then more pieces get cut off, and then it finally becomes the active peptide. And that's what's going to bind to the receptor on the target cell. So these protein messengers are going to be stored in vesicles, and they will be released from the secretory cell by exocytosis. And this is just showing the synthesis of insulin, one of the protein hormones. So we're going to start off much larger protein, this pre-proinsulin up here. And then we're going to cut off this blue sequence. And now we're at proinsulin. And then we cut off kind of this tan sequence. And now we're finally at the active insulin. Okay, so the protein messengers get made as a larger protein, pieces get cut off, and then finally it becomes the active form. And then just a reminder that these protein messengers, the directions on how to make them, come from the genes in your DNA. So for example, there's an insulin gene in your DNA. And so when it's time to make insulin, you'll transcribe that gene, you'll copy it, make the messenger RNA, and then you'll translate the messenger RNA with the ribosomes, and we're going to be translating it on the rough ER. Things that are translated in the rough ER, they're going to be packaged into vesicles and then secreted by exocytosis from the cell. So that's why we go to the rough ER there. And so we make it in the rough ER, package it in a vesicle, it'll go to the Golgi apparatus where it gets modified further, and then stored in these secretory vesicles until the pancreas cell gets the signal that it's time to release insulin, and then it'll release the insulin by exocytosis. And this is just showing the formation of insulin. So you can see the ribosomes on the surface here of the rough ER. They're translating the messenger RNA, making the pre-proinsulin, and then part of it gets chopped off here on the way. and package it up into our vesicles, send it to the Golgi apparatus. We do some more modification, and then finally it becomes the finished peptide messenger or protein messenger, and that's the active form, and then it gets the signal to release it by exocytosis. The next chemical class is steroid hormones. Steroid hormones are all made from cholesterol. And remember, cholesterol is a type of lipid. And remember, lipids are all hydrophobic. So we're going to find the target cell receptors inside the cell. So they're going to be in the cytoplasm or in the nucleus. And because they're hydrophobic, they can't be stored in vesicles because they just diffuse right out through the membrane. So as soon as they're made, they get released from the cell. And how they're going to be released from the secretory cell is by diffusion. So they just diffuse right out the cell membrane. Some of the steroid hormones, which we're going to be learning about in Chapter 6, we have estrogens, progesterone, testosterone, so all of the sex hormones. We also have cortisol, stress hormone, and then aldosterone. We'll talk about what that one does later, and also vitamin D3, which helps regulate your calcium levels. And this is just showing the structure of cholesterol. Remember, it has the 4 rings that are fused together. And from cholesterol, you can make all of the steroid hormones. So here it's just showing making cortisol, and then some of the sex hormones, progesterone and androstenedione, which can be turned into estrogen or testosterone. And I think this is a really interesting picture showing the synthesis of the steroid hormones. So we're going to be starting with cholesterol. And where we get cholesterol from is from our diet, and also remember the liver can make cholesterol as well. And so as we follow the pathway down, we can take the cholesterol and we can certainly make progesterone with it. And then from progesterone, we can make the cortisol and aldosterone. And then also if we go down this other pathway, we can end up making androstenedione, which can be used to make the estrogens and testosterone. And then what I think is just fascinating is that testosterone can be used to make estrogen. So in women, we do make testosterone. We leave a little bit of it as testosterone, but most of it we convert into estrogen. Whereas men, they do make a little bit of estrogen, but they're going to leave most of their testosterone as that. The next chemical class of messengers is eicosanoids. Eicosanoids are a type of lipid, and would these be hydrophilic or hydrophobic? Hopefully you said that these are hydrophobic because they're a lipid. Eicosanoids are made from the fatty acid part of the plasma membrane phospholipids. So the phospholipid is going to be broken down, and those fatty acids will be made into arachidonic acid, and then the arachidonic acid will be made into the eicosanoids. Where the eicosanoids got their name is eikos. That part means 20. And eicosanoids have 20 carbons in them, and so that's where the name came from. And there's a number of different types of eicosanoids that we're going to be talking about. We have prostaglandins. There's a lot of different types of prostaglandins in the body, and they have lots of different jobs in the body depending on which type they are. But just one function I want you to remember of the prostaglandin is that some of them are involved in inflammation. The leukotrienes are made by some of the white blood cells. Leuko means white, so maybe that'll help you remember that. And the leukotrienes are involved in inflammation. Thromboxanes will help cause the blood to clot. So if you remember, a thrombus is a blood clot. Hopefully that'll help you remember that one. And then prostacyclins will do the opposite of the thromboxanes. So prostacyclins will prevent the blood from clotting. All right, so knowing that eicosanoids are hydrophobic, where do you suppose the target cell receptors are going to be located? So would they be on the cell surface, or are they going to be inside the cell? So hopefully you said that they're going to be inside the cell. So remember that eicosanoids being hydrophobic, they can just diffuse right through the cell membrane, and so they'll bind to receptors that are inside the cell. Eicosanoids are going to be synthesized when they're needed. They cannot be stored. And the reason that is that remember that if chemical messengers need to be stored in the cell, they would be stored in a vesicle, little membrane-bound sac. But because they're hydrophobic, they would just diffuse out of the vesicles, so they cannot be stored. So as soon as they're made, they just diffuse right out of the cell. So they're going to be released by diffusion. So they just diffuse out of the cell. And all of the eicosanoids are going to act as paracrines. And remember that paracrines are just going to diffuse out into the extracellular fluid and go to nearby cells and just talk to the neighboring cells. So they're not going to go very far. This picture from your book shows how the eicosanoids are made. We start at the top. We've got a membrane phospholipid, which is going to be broken down by an enzyme called phospholipase A2, which you do not have to know. This enzyme will break the phospholipid down into arachidonic acid, which can then go down two different pathways here involving some other enzymes, and then make the eicosanoids. So the two pathways, we've got the cyclooxygenase pathways, which use the cyclooxygenase enzymes, which we're just going to abbreviate as COX, and there's two enzymes, COX-1 and COX-2. And then the lipoxygenase pathway, which we'll just abbreviate that enzyme LOX. That enzyme is going to make the leukotrienes. So we're going to just concentrate on the cyclooxygenase pathway and take a look at these COX-1 and COX-2 enzymes and see how we make these prostaglandins, prostacyclins, and thromboxanes. All right, let's draw a picture of the cyclooxygenase pathway. And the reason that I want to focus on this pathway is because there's a group of drugs that actually inhibits the COX enzymes. So this group of drugs is the NSAIDs. So that stands for non-steroidal anti-inflammatory drugs. So this would be, for example, aspirin, ibuprofen, naproxen, Celebrex. Those would be examples of NSAIDs. All right, so let's take a look at the cyclooxygenase pathway and just show you how this works. So we've got our membrane phospholipid. We're going to break it down into arachidonic acid. And then we've actually got two different COX enzymes. We've got COX-1 and COX-2. So the COX-1 enzyme is the good enzyme, and the COX-2 enzyme is the bad enzyme. So the COX-2 enzyme is the one that we do want to block with our NSAID. So the COX-2 enzyme is going to produce from arachidonic acid some prostaglandins that will cause pain and inflammation. So like if you sprain your ankle and you take some ibuprofen to help with that pain and inflammation, that ibuprofen is going to be blocking this COX-2 enzyme, and then you won't be producing these prostaglandins, and then that'll decrease the pain and inflammation from your sprained ankle, and then you feel better. Okay, so blocking COX-2 enzyme is good because you block the pain and inflammation and you feel better. Okay, so we do want to block COX-2 with our NSAIDs, so that's actually good. But the problem is that our NSAIDs will also block the COX-1 enzyme. And the COX-1 enzyme is actually the good enzyme, and we don't want to block COX-1. And when we do block the COX-1 enzyme, that's where we start getting our side effects with our NSAIDs. So let's show you what happens here. Okay, so our COX-1 enzyme will take the arachidonic acid and will produce the eicosanoid thromboxane with that. And remember that thromboxane will cause the blood to clot. Okay, so if you're taking an NSAID, one of the side effects we could see is that it could cause bleeding, which could be a problem if you're going in for surgery. Because the NSAIDs will block the COX-1 enzyme, you don't produce enough thromboxane and then you don't clot your blood as well. So you could have excessive bleeding during surgery, which could be a problem. But we could also use that to our advantage, because if you have a person who's had a heart attack or a stroke, we actually tell them to take a low dose of aspirin, because we know the aspirin is going to block that COX-1 enzyme. They don't make as much thromboxane, and then they don't get, they're not as likely to form the blood clots, and then that decreases their risk of having another heart attack or stroke. So that's why we recommend aspirin for those patients. Okay, the other thing the COX-1 enzyme will do is it will take the arachidonic acid and you'll produce some prostaglandins. And these prostaglandins will increase the mucus in your stomach. And that mucus layer in your stomach is actually important because it protects your stomach from the stomach acid. And the prostaglandins will also decrease the amount of stomach acid that you're producing as well. So that's all good, okay? So the COX-1 enzyme will also do that. So another side effect we can see with taking these NSAIDs is that by blocking this COX-1 enzyme, you block the production of these prostaglandins. You don't produce as much of that protective mucus layer in your stomach. You also end up producing more stomach acid, and then you can end up with a stomach ulcer, which could be really bad because the ulcer, if it ends up going all the way through your stomach wall, then you have stomach contents leaking out into your abdominal cavity, and that can actually kill you. So that could be pretty serious. Okay, and then the last thing that the COX-1 enzyme will do is it produces some other kinds of prostaglandins. Oops. That are in the kidney, and they increase the blood flow to the kidney. And kidneys like to have a lot of blood flow to stay happy. So if you're taking NSAIDs and you block this COX-1 enzyme and you decrease the production of these prostaglandins, that are increasing the blood flow to the kidneys, that'll decrease the blood flow to the kidneys. And then that can make the kidneys unhappy. And especially if you already have some pre-existing kidney problems, that can make those worse. So that could, push you over the edge and put you into kidney failure. So you have to be careful with patients that do have some pre-existing kidney problems. NSAIDs may not be the best choice of drug for them. Okay. There are some of the NSAIDs that are more selective in that they don't inhibit COX-1 as much. And those would be things like the Celebrex. They've designed that drug so that it's more specific in that it's going to be more blocking COX-2 and not so much COX-1. And so you don't see quite as many side effects with the Celebrex. I mean, there's some other problems you can have with it, but you don't see as many of these other issues as you would like if somebody's taking ibuprofen or naproxen or aspirin. So, but just, you probably have heard of some of these issues with the NSAIDs, and you'll see this again in your healthcare classes too. So, all right, this is a really good table to know from your book. This shows all of the chemical classification of the messengers, and make sure you know whether they're hydrophilic or hydrophobic, and also know where the receptors are on the target cells. So are they on the cell surface in the plasma membrane, or are they inside the cell? I'm not going to ask you this last column over here. You don't need to memorize that. Okay, I won't ask you that. And then table 5.5 from your book is also really important to know. So this shows you hydrophilic messengers and then hydrophobic messengers, and just compares the two side by side. So I would definitely spend some time studying these, because you can see me writing some test questions, knowing the difference between the hydrophilic and hydrophobic messengers and how they work, the differences, which chemical classes each one is, how they're transported in the blood, where receptors are located, how they're released from the secretory cell, how fast the response takes. So there's all kinds of stuff I could ask on from this table. So that would be important to know too.